bioinformatician and lab manager at MSU's Kellogg Biostation. Tyler's work has focused for a long time in developing molecular, conceptual, and computational ways to do population genetics with next generation sequencing data in non-model systems, and I think that is something a lot of us here struggle with. Tyler did his PhD at UC Berkeley with Rasmus Nielsen and Monty Slatkin. He then moved to Cambridge to work on cichlids with Dick Durbin in Cambridge, and he's currently at MSU as you all saw. And yeah, I'm not gonna
take it up.
Talk anymore.
Cool. Let's see. Wow, thanks a lot. I'm really pumped to be here talking about something that I feel like I haven't been like really passionate about work-wise in a while. Not that the cichlids weren't cool, but like trying to save something here, which I think is really neat. So thank you for having me. And today I'm going to talk about a little work. I arrived at KBS, started a little over half a year ago, so in April, so 7 months or whatever it's been. And this is what's come out of it so far. I'm pretty excited to share. So, I mean, when I look at the world we live in, this is what really motivates me to do what I do, both on a personal level and a professional level. And it's just, I look at these landscapes and I think, you know, how do populations persist in them? And sometimes it's hard for me to even wrap my head around because, you know, I think this type of fragmentation, this type of rapid change, I mean, it's often to our benefit. So it's sometimes hard to relate, you know, how hard you're making it on, you know, all the organisms that we share this planet with. And so it motivates me personally because I've always been interested in wildlife, like animals, plants, all that. At a professional level, then it brings me to, you know, really making me curious about what can conservation science do to aid populations in persisting in these changing landscapes, in these fragmented environments, and specifically how to keep them out of this extinction vortex that you get when populations start to become very small and start to become more susceptible to demographic and environmental stochasticity, such that when, you know, a freak occurrence happens, then the population is even smaller, then they become more susceptible to inbreeding, and maybe their fecundity or viability will drop, and the population's getting smaller, and that demographic buffer gets smaller and smaller. And so you start losing your genetic and your demographic buffers, and it's bad news bears. And so when that, you know, you want to keep that from spiraling out of control. And so one effective way to pull a population back from this or prevent them from entering it is through gene flow. And of course, that's becoming a big problem in these more fragmented habitats. And so if you have a population that's declining, it's been shown, you know, if migrants can enter into the population, it can rescue that population. By rescue, I mean they undergo a demographic increase, so the population size starts to increase, the population growth rate increases. And also, hopefully you also get this genetic rescue where inbreeding decreases, and hopefully you get an increase in genetic diversity. And so what I'm particularly interested in is how genomic data can inform on how better to use gene flow to promote the persistence of populations. Because a commonly used technique around gene flow in these fragmented habitats is assisted migration, whereby we're going to move individuals from different populations around to try to create gene flow. And I'm interested in how can genomics be utilized to improve these assisted migrations. And we know they work. There's been great examples of this in many iconic cases whereby populations are on an extinction trajectory. Their populations are declining, and individuals get introduced, so translocated into these populations where these pink arrows are, and then you see the rescue effect, this increase in the demographic, in the population size. And, and there's a lot of focus on this and considering that being a successful rescue is this increase in the population size. And what I kind of wonder about is that's, of course, really, really important. That provides the demographic buffer. That's essential. But what is also happening at the genetic level? And so does this lambda greater than 1 really fully qualify as a success? Or should we be considering, is it important to look at the genetics? Or can we just assume that by proxy of the population size getting bigger, our effective size is also increasing, et cetera? And so we have the opportunity to look at that in these in Florida scrub jays, and so that's what I'm going to be talking about today. And a little background about their unfortunate history is they've suffered massive declines. So they're very reliant on Florida scrub habitat. They're endemic to Florida and non-migratory, and very reliant on this scrub habitat there that has, for the most part, been destroyed. There's only 10% of it left of what it was historically, and which has caused the populations to undergo a massive decline, and leading to their federal listing as threatened in 1987. And a little bit of background about the characters of this story, the things that make them interesting, perhaps a bit challenging for management, is they're very reliant, like I said, on this particular type of habitat, this xeric oak scrub. And they're strongly philopatric and territorial, so they don't disperse well, which, like I just said, gene flow is important. If you don't disperse, it's a pain. And to try to help them out. And they're also cooperative breeders. So usually that means there's one monogamous pair in this kind of family group comprising helpers. Those are usually the children that help with food provisioning and defending territories. And so like you can see some ideal scrub habitat right here. And so that's what a lot of habitat restoration efforts are trying to create. And so I'll be focusing for this talk on this West Coast population called the M4 metapopulation. And Mosaic Fertilizer, which is this phosphate mining company, owns territory in the M4 metapopulation. And so they've been involved with the Fish and Wildlife Service to develop this habitat management plan to aid in the recovery of the Florida scrub jay. And so that's who we've mostly been working with on this project, is Mosaic. And so there were population viability analyses conducted, which ultimately concluded that the best way to aid in the recovery of the M4 metapopulation, or the persistence of it, I should say, is to change the spatial structure through translocation. So it was ultimately considered to be in this have this non-equilibrium spatial structure, these small fragmented populations. And what they want to achieve through translocations is generate this, what the PVAs predicted would be a much more stable mainland island kind of spatial structure, whereby you have this big island mainland that could serve as a source to these more satellite populations. And so to achieve this, they translocated, the idea was to translocate birds into this geographically central area right here called the wellfield population that is sitting right adjacent to a public lands, a preserve. So it was ideal because it presented new habitat and it had been recently restored. And so it would be an ideal place to establish this mainland island. And so just prior to the translocations, there was only 4 family groups consisting of 13 individuals there. And so the main goals of this recovery, or this whole plan, was to preserve these lineages that existed in these particular subpopulations in areas that were going to be mined. So they were surely going to face extinction. And so to preserve these lineages, they thought, we'll introduce them into the Mosaic wellfield. And also, by doing that, we'll rescue this small, isolated population. And then that would ultimately lead to the help aid in the persistence of this M4 metapopulation. And the translocations and all the initial recovery efforts were carried out by Raoul, Lauren, and Reed. And then Sarah and I have more recently come on the project. And so what happens is that from 2003 to 2001, 51 Jays in total were translocated from various, let's see this, what is this, five subpopulations outside the well field. So they were all taken from these sites and introduced into this recipient site here at the well field. And I'll kind of refer to the well field in the Duet Preserve. I'll just call it the well field population. And as you can see, after birds were introduced, after these introductions, you could see the population beginning to demographically expand. So you start to see this. These dots are family groups. So you start to see these family groups, new groups crop up and spread throughout the well field. So it looks fantastic from that perspective. And indeed, when you look at the demographic expansion, it's quite clear. This is very reminiscent of those plots I was showing before, when translocations had been carried out into these kind of threatened populations. Whereas individuals were introduced here by these pink arrows, and as you can see, the number of family groups by these bars was increasing, and also the overall population size was going up and up and up and up. So from a demographic perspective, it was a success, I would say. The population definitely grew. And the growth rate continued to increase. But then, like I said, we were interested in, so what is happening genetically? What's going on under the hood of this expansion, basically? Is it all groovy as well? Like, did we not have to worry about, was this a success and the well field's gonna be fine? And so in order to do that, we did some whole genome sequencing of 87 Jays total to about 7x depth from resident Jays from the well field. And those were Jays that existed in the well field prior to translocations, just prior. And then also sequenced translocated birds, 30 of the 51 translocated birds. Some individuals that were from around the same time period of the residents existing in other kind of M4 populations. And then about in 2021, resampled the contemporary wellfield population. I should say it was sampled in such for a population genetic analysis, so to kind of minimize the relatedness. So one individual per nest, I believe, from every nest in the well field. And there was definitely a genomic change following these introductions. And I should thank Nancy Chen right now, who provided the reference genome and kind of helped enable a lot of these genomic analyses. So based on these little over 2 million SNPs, you can see a clear genetic change from this PCA here, where the resident populations are these dark purple Rs, and the contemporary populations are these pink Cs. And so you can see the contemporary population has diffused, across both axes of this PCA, in which the residents are kind of nested within. And it's quite a drastic genetic shift. This is about, since the introduction started, it was about 4 to 5 generations, scrub jay generations. And you already get this, and the population diverged genetically by about 3%. And another thing we looked at, though, was try to estimate this genetic skew parameter. So it's a measure of genetic contribution based on relatedness, which we calculated from the genomic data. And it kind of considers 2 factors. It's kind of the proportion that any one of the potential founders, so these are all the translocated and other M4Js, the contemporary individuals aren't here. So it's kind of encapsulates the idea of the proportion of the contemporary population that any particular individual is related to above a certain background relatedness level, because there was background relatedness. in the population. And so we're interested in kind of what relatedness could occur post-translocation, only this recent kind of fueling of this expansion. And then it also considers the probability that any particular of these potential founders is more related to sort of a randomly chosen contemporary individual than any other founder. So those are roughly the things that it considers. And as you can see, we see very strong skew, very strong unequal genetic contribution, especially by the site 1 T71 bird, a site 18 T99, and then it kind of starts to flatten out. And so it looks like we have very high or very skewed contribution, both among sites where it's only really site 1 and site 18, and also within sites based on the number of on the individuals that are actually contributing. And I should say, based on how this is calculated, since it's kind of a relatedness-based measure by proxy of relatedness to a particular individual, like these have high relatedness to that individual here, that other site 1 individual, they're going to show up as having high skew. But because of the second factor thing, that this probability of being more related than another individual, it's likely that the individual with the higher estimated parameter is the actual direct descendant. So we can kind of ignore these because they've just been pulled up by proxy of high relatedness to that individual. And you can kind of see that if you look at it another way and really want to cross your eyes and boggle your brain. If you just look at this sorted relatedness matrix, that's all it is where we have the contemporary individuals here, and it's just been sorted by pairwise relatedness, which descends as you go up. So as you scan across all these contemporary individuals, as you go back and you're seeing the individuals they're most related to, and under this black dot is all this relatedness kind of below that background level. You see, you start hitting, okay, T71, 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 and a lot of them, and that's that T71 bird. And that's kind of what's being factored into that skew parameter. And then if we look at that in terms of, the proportion of the sample that these individuals are related to above the background level, you see that this T71 individual and this T99 individual, they're related to, at least more than half, more than twice as many individuals in that contemporary sample than any of the other potential founders of this expansion. And it's quite high. So the T71 individuals closely related to 43% or 12 of those contemporary individuals in our sample. Similarly, T99 is related to 39%. So, and that's actually what this PCA is showing, right? Which I think is a good lesson, because, I think maybe my first knee-jerk reaction when I saw that, it's like, okay, this diversity is nested within the resonant diversity is nested within the contemporary. Maybe, it's because that's a genetic subset, a smaller genetic subset of the contemporary diversity. But I think it's more just, you're seeing this effect that, the population, different parts of it, are becoming much more like the individuals they're descended from, right? They're just being drawn towards their ancestors in that population. So what's the consequence of that? I mean, hypothetically, in these genetic rescue scenarios, what you're hoping for, the goal is that it should, if we're comparing the contemporary to the resident population, from the resident population to the contemporary, it should drop inbreeding. That's what you're trying to achieve. And likewise, heterozygosity should go up. But we see the exact opposite, right? And that's what this skew is doing, that a lot of this population, this new contemporary population, is being descended from just a couple individuals. So now actually we see that heterozygosity has significantly decreased, and that's also, makes sense as that inbreeding has significantly increased. So actually, genetically, not what we had hoped for. It's not. Is this a problem that now this well-field population is on a trajectory of increased inbreeding? I mean, granted, there hasn't been much time, right? So keep that in mind. That time will could tell what could happen, but it's worrying because that inbreeding is decreasing or increasing, because that is a problem in scrub jays and other species. In scrub jays, Nancy Chen has shown relationships between as individual inbreeding coefficients go up, you get reduced viability, and likewise, as kind of also inbreeding increases, you get increased hatching failure. And probably the most iconic and famous example of where this skew is really a problem is the Isle Royal wolves. I always grew up calling them Royale, but a native Michiganer told me it's Isle Royal. So, I'm just throwing that out there that I've been told, I've gotten mixed signals. And I'm going to say Royal because someone who I think knows told me. In which case, you had the small population of wolves that had colonized Isle Royal and became inbred, and then you got this immigration of this one individual later, I think in 1997 or something like that, and they was behaviorally dominant, and there was initially this rescue effect because that population was kind of inbred before this new migrant showed up. But once he showed up, so there's this kind of initial rescue effect, but then in 11 years, nearly 59% of that population drew their ancestry from that one male, and then that quickly led to inbreeding depression. Now the population has collapsed. And so what we're seeing, is reminiscent, I think, of this, where we have high skew. We have a couple individuals that look like to really be fueling this expansion, and hopefully we don't. And so this is something to be careful of. And so, it would be nice to understand what, for these translocations, what are there features of the translocation itself that led to this happening? Was it a function, let's say, of just the time since translocation, so the amount of time that a bird had to be there. So this is the time since the first introduction, and here's this genetic skew. And of course, we're trying to these kind of trends are fit to try to include the independence among these points that we're fitting these to is the most likely contributing individual from these closely related family groups. But this chronology of introduction doesn't seem to explain. I mean, it could also make sense because the first birds introduced would have the most available habitat, which this chronology is pretty much a good indicator for. But it doesn't seem to be at all related to that. And there's many other factors to consider. So the age or experience of the breeding pair, things like the family size, the conditions that year, what season, what kind of resources were available to them when they were introduced, the physical condition of the individuals. We have kind of all this data, and it's things that we're starting to consider and look at. But I think it could also just be a very intrinsic quality. It could be like the null model, a very intrinsic quality of cooperative breeding animals that could have been a very evolutionarily stable and great strategy for them in a more contiguous habitat where you had more migration, where particularly fit individuals contributing largely to a population could keep could produce many more fit individuals, but you wouldn't have this inbreeding effect because there was more historically migration to counterbalance that. And that's just an equilibrium, or so quote unquote, that is, I think, difficult to achieve basically in a more fragmented world. And something that might speak to this being the case is we see a very similar, we just started collaborating with Reed on this red-cockaded woodpecker data set. And you see something eerily similar to the mosaic translocations, where the population had been declining, and then you had translocations begin here to try to combat that, to try to rescue the population. And it did start to go up, but then you had this big hurricane. And it's probably actually could be important that they started these translocations and the population started to increase, right? Because that provides the demographic buffer, and that's really important to have some resistance against these this environmental stochasticity. And so maybe it gave them enough buffer that they needed to then still rise after that hurricane. But very much like the mosaic, very much like Isle Royale, now 33% of the population is completely descended from one male who just had large reproductive skew. And along with that, you see inbreeding also increasing throughout that population. And now there's actually been some indication that it's some very early subtle indication that maybe perhaps there could be some inbreeding depression starting to occur, whereas right now its hatching failure rates have plateaued out in this rescued population, actually above what they were prior to the rescue. So in light of all of this, how can the odds for long-term persistence be improved? Like, kind of what have we learned from this? And I think it says harking on how important monitoring is. I mean, this is the lesson that we learned, is that, I would say seeing the demographic expansion isn't enough. Like, this could happen. And the way you can detect it is through monitoring, and particularly genetic monitoring. That's, it speaks to the importance of that, whereby, by using genetic monitoring, you can detect early these signs of skew, and that your population might be at increased risk of inbreeding depression from skew if it's occurring. And the great thing about monitoring is that, I realize that monitoring populations is a really timely endeavor and expensive, logistically difficult. And probably the great thing about genetic monitoring is you don't have to do it every year. You only need an occasional snapshot to get the resolution that you would need to be able to detect these things. I think that demographic modeling or monitoring is also continuing. That is also very important. But again, in thinking about how genomic data can improve those efforts, it can definitely, in these very time-intensive efforts, it can definitely tell you what's worthwhile measuring or what to look out for. So, case in point, for example, one kind of, if you might make the assumption, and I think a common assumption in this monitoring is that for birds, like, the number of total fledged offspring should be a pretty good indicator of that bird's contribution to the population. But again, if we look at the total number of fledged birds and relate it to this kind of genetic contribution, this skew parameter, there is usually agreement. So again, like for T71 and T99, they did produce quite a few offspring, and they genetically, their ancestry is all over the place. But the problem is that there's definitely outliers, and so it's not a foolproof method. So if you look at this Texaco bird and other birds from Texaco, they produce some of the most offspring that we see, but their ancestry isn't anywhere to be found in the population. So that's important to consider, because that's a whole nother lineage that you might think is represented in this population, and it's not there at all. This would tell you, if you're monitoring, I'm not going to use total fledging as a foolproof indicator of who's actually contributing to this population. It's probably important that you should invest time in, assessing breeder recruitment and things like that. Another great thing about having genomic data, of course, is that it's really great for enabling demographic inference. So you can infer the demographic history of these populations. And in these translocation scenarios, which I think this, is saying it warrants, is continued translocations, that it kind of gives you a target. You can you can assess more, historic levels of migration in this species and try to emulate that through your assisted migration. And so you can fit these kind of models with genomic data. And a big point I want to say is that, I think that what we learned here is to sustain the mosaic wellfield population, additional translocations are probably quite important. We need that kind of additional migration into this population to combat this increased inbreeding. And so it's not over. Despite that it looked like it was a success, and we could call it done, that the mosaic is well off, this is probably a call for no, translocations need to keep happening at maybe, an informed rate. And lastly, I think this is also calling for a lot of policy surrounding translocations, and that both demographic and genetic status should be assessed. So for example, the Florida Scrub Jay Recovery Plan, the delisting emphasizes both population size, so that kind of demographic size, and genetic diversity. So genetic diversity has to be naturally maintained without translocations. In the red-cockaded woodpecker, however, it explicitly states that populations become ineligible for translocations after they achieve a certain population size, so 30 potential breeders, which means like in the scrub jays, they would already be ineligible. So you would not be able to translocate new jays into this population to try to combat this inbreeding. And that's a kind of, in my mind, a flaw in some policy that needs to be reformed, because they're probably experiencing very similar genetic effects as the scrub jay. So in sum, was it a success? I would say in one way, yeah, demographically, and that's important. This is not an anti-translocation. I think the translocation should be done. I just think we've learned a lesson from this, and that you need to continue to monitor, and you need to importantly look at what is going on under the hood of these translocations. So yeah, we've achieved this demographic buffer, or at least an increased one, but there was no genetic rescue, and actually quite the opposite. So to achieve this model, a healthy mainland populations, I think continued translocations, as I've been harking on, are probably warranted. And again, I just wanted to emphasize this isn't an anti-translocation talk. It's just, we should recognize that this can happen to avoid potential island royales, and we do this through continued monitoring. And I just want to say, it's exciting where I really think all this translocation work is really fascinating, and we're studying populations on the other coast of Florida, like Natasha Lear, and collaborating with Tram at Cornell to look at, the effects of translocations in this population, kind of come up with some landscape genetic perspectives, insight into the metapopulation dynamics, and hopefully wanting to relate some genomic features to population vital rates, things like that. So just to plug that, it'll be interesting to compare and see, are we going to see Skew in these introductions as well? And now I'm going to quickly transition to something that is much, much more conceptual at this stage, and is what I've been thinking about in what could be a useful way to take genomics to aid in species recovery. And I'm going to be talking about the Poweshiek skipperling here, and I should thank Dave Pavlik, who's a research assistant in the Haddad lab at KBS, who's kind of carrying the weight of this species on his shoulders, really. He's done incredible stuff, and also gave me a lot of materials, so that's great. But the Poweshiek skipperling is this historically abundant species that you find in the northern Midwest, and just right in the southern tip of Manitoba. And it's a very reliant species on tall grass prairie and prairie fens. So this shaded habitat is kind of the historic extent of the suitable habitat, and these black dots are the kind of historic sightings for this species. And now they're effectively, very close to being gone. And so you only find them in these two, where these green arrows are, two spots. So one population that's preserved in Manitoba, and at a handful of sites in Michigan, and that's kind of it. So things are looking pretty grim, and actually just recently, this last field season, you could almost find none in Michigan. And so the threats to them have, again, been habitat destruction, recurring theme, pesticides. They think that climate change and hydrological changes could be to blame, and they've also could have been experiencing kind of these effects from that extinction vortex as these populations have become quite small. And so the declines that started occurring in the 1990s have ultimately led to this crisis and led to their listing as endangered under the ESA. So they're starting to, so they have federal protection. And there's a lot of excitement for their recovery. So there's now captive breeding programs at the John Ball Zoo and Minnesota Zoo. There's also recovery efforts in Canada. And so they capture wild individuals, they breed them in captivity, and then they re-release them. There's habitat management going on, surveys to monitor them. And like right when I got to KBS, there was this big ribbon cutting ceremony for the new breeding facility at John Ball Zoo. It kind of fired me up for these butterflies. It was cool. But there's also, in these captive populations, there's definitely look like signs of inbreeding depression. So the caterpillars are failing to develop. Sometimes they're too weak to emerge. There's some pretty clear physical deformities. And so now, again, Fitz and I have been brought in to tackle kind of the genetic aspect of this recovery. And so I'm interested in, one thing that needs to happen is we just need a good old-fashioned population genetic characterization to so understand what the inbreeding levels are, kind of the structure in this population. One thing I'd like to do is be able to try to understand the loci underlying these phenotypes manifesting through inbreeding depression. And kind of to this end, we just started this, so we've currently just sequenced 41 of these Poweshiek genomes. But what I want to focus on really here with these last few minutes, I'll go through quite quickly, is a conceptual framework that I've been thinking about to be able to do this. And don't get too invested to the Poweshieks, because this has to take you back to where like this idea was of how to do this was spawned. And that was in my postdoc when I was working on studying the diversity of the Lake Malawi haplochroma cichlid, so that quite famous adaptive radiation. And so what I was really interested in, I was lucky enough to kind of get to generate and analyze this pretty impressive data set consisting of over 2,000 whole genomes representing 255 species that span all that genetic or phenotypic and ecological diversity. And I spent a lot of time, in doing GWAS in this lineage, GWAS in that lineage, a lot of divergence mapping. But what I really want to do with this quite unique data set is try to figure out how to leverage that information to figure out, is there a way that I could identify any kind of instrumental or key genes that have underlied this, burst of radiation? Kind of like what genes make a Mbuna, or what genes make a Ramphicromus? And long story short, kind of at the end, my aha moment is the key to doing this really seemed to lie in my experience in these biclustering algorithms. So they're algorithms where you're essentially trying to jointly reduce the variance between kind of the rows and columns of a matrix. So minimize that mean square residue between them. And that kind of can tell you how allele frequencies are fluctuating in unison across species. So in my case, I had kind of loci along the columns and loci along the rows and species along the columns. And this was, I found to be quite effective for finding candidate, these sets of suites of candidate loci, and they revealed quite interesting loci. It was useful for many regions. It's also quite effective at finding these structural variants, so like inversions that seem to be playing a big role in the diversification. And a great thing about these biclustering algorithms, it's kind of an exploratory tool in a sense. So I was able to identify a lot of traits seemingly important to different kind of levels of this radiation, evolutionary levels of this radiation that I wouldn't have thought of a priori. For example, it seemed like I finally started to realize that these heart-related genes were really important to this Mealandia complex, so the development of the heart. And another thing that gave me strong confidence that this wasn't just giving me garbage is I could throw out many different kind of independent tests for signatures of selection at these loci and they would tend to stand up against those. So they showed, then signatures consistent with selective sweeps, things like that. So what I'm wondering is, can biclustering be used to pinpoint these suites of loci underlying these traits related to inbreeding depression? And I should say that I'm kind of working on the assumption that, we're considering inbreeding depression as a result of, many recessive deleterious mutations being expressed in various combinations, but a whole suite of them. So whether that assumption holds or not, we'll see. But in here, I'm not gonna go really into the method, but kind of walk you through conceptually what I'm working on, basically. And so if you look here, I have this simulated Poweshiek genome. I should say one of the things that biclustering was really useful for, I think its most common application in omics, is identifying gene co-expression networks. So if you kind of think of the genotype as being this expressed quantity or this allelic dosage, you can maybe find these suites or like networks across the genome, right? This latent structure. And so if I simulate this in a very oversimplified simulation, right? Hundreds of thousands of SNPs, and then have individuals here. And then now my quantities are the dosage of a derived mutation. What you might expect to see in a small population, a small bottleneck population, is increased drift. So if you scan in and get some little better resolution in the region, you can see sites where the derived mutation has drifted to high frequency in many of these situations. And I should say right now, I'm not modeling linkage or something. This is way oversimplified, but also helps in the visualization. And so let's say we imagine this imaginary set of traits that I'm assuming are polygenic and overlap in some degree in their polygenic basis. So for example, we could say something related to development. So, that we would monitor for in these captive breeding programs. So like deformed wings, deformed exoskeleton, even low fecundity, because that might point to developmental issues with their reproductive system. And for all intents and purposes, for the resolution that we might want to look at, we could consider anything showing these developmental abnormalities as cases and healthy individuals as controls. And here I've just reordered those rows so the individuals, and kind of clump them together just to make the visualization easier by kind of their phenotype, their imaginary phenotype. And then if you knew the loci, underlying these traits, or the mutations, the sites where these mutations are affecting these traits, which I'm assuming I do, I'm assuming I know the truth to show you what, you would see, is the structure becomes quite apparent. So these are SNPs where across them, if they tend to start expressing these deleterious mutations in concert, you start to see them propagate again across these different phenotypes. And the phenotype might be different. It'll depend on, if they're polygenic, the genotypes at other loci that might not be in the subset. So you might not always get deformed wings, but some developmental aberration. And so, but this is the problem. The structure is very apparent if I show it to you, right? But can you extract that from the genome? And so if I run this biclustering algorithm that I've been working on these, you can recover that latent structure. And, a nice feature of this is it has, statistical things where you can test for these trait by SNP interactions through permuting this matrix to test for, the significance of these biclusters. And you can, so I have been able, in this very oversimplified scenario, right, been able to pull out that latent structure, which this is way oversimplified, but if it doesn't work here, it won't work in a more realistic scenario. And another thing that's cool is, this identified the case control bicluster, right? But you'll also be able to discover secondary biclusters, so subsets amongst the traits even, which could allow you to differentiate different loci affecting different aspects of development, or things or loci that affect viability versus fecundity, for instance, or features of fecundity. And so what I'm seeing where this could go through in an association, like the grand vision, there's not a lot to lose by trying new things with these butterflies. They're in pretty dire shape. So what I'm proposing is that we can develop almost a screen for, if we can map these suites of loci, we can develop a screen for them that we can implement in a subset, at least, of the individuals in this captive breeding program. I mean, I wouldn't propose that we start doing it everybody, but we try it in some, and we compare the fitness of the individuals that we get out if we screen for these type of associated loci. And so, the idea is we could design these crosses that minimize the chances of these associated SNPs being expressed in their kind of homozygous states. And these are individuals being released into the population. Ideally, if it was looking like it was working, this could mitigate the spread of these detrimental mutations and suppress them in the wild population. Of course, do this with a lot of caution, right? This is kind of conceptual idea for conceptual idea right now. We would want to be very careful for trying to use it. But, again, this is still a huge work in progress. And like this whole methodology and thinking about how to do this. So, right now into the algorithm, I'm thinking about how to incorporate physical linkage. Like, it might not be that important. Like, if we're just trying to screen, it's not like in the cichlids where I try to map a particular causal locus and, do the CRISPR experiments to see, to really validate that as a causal thing. Even if we're detecting causal SNPs by proxy of linkage, if we can screen for them, I think that could still be useful. But I'm thinking about how to incorporate physical linkage. And also, I think the pedigree information will actually be quite important. So figuring out how to do that. And of course, then at this point in the development of this idea and the stage, I have many more simulations and making them much more realistic to assess this. But I wanted to share this because this is kind of what I'm thinking about, and this is one way I'm thinking how can we actually leverage genomic scale data in the recovery of this species. And I think there's a lot of information there, and identifying this polygenic structure I think would be a powerful tool, but I'm trying to think about how to do that, and this is one idea. So that's all I've got. And so just thanks a ton for listening and attending this, and thanks to all the supporters. So yeah, thanks a ton.
I have some time for questions, so wait.
Oh, you'll start here and work our way back, yes.
I really like the simulation I did, the theoretical framework you laid out. I was wondering, because it looked like you had made each loci either associated with the phenotype or just not at all associated. Yeah. And I was wondering if you were thinking of, because a lot of times we find that things are contribute 1% of the variation or something, and if you were gonna try and do it at a more continuous
Yes.
scale.
Yes, ultimately. I mean, I think that will propagate through kind of different I'm imagining, for this to work, there has to be some suite, some structure there. If there's no structure to discover, so I mean, there's hopefully many different subsets, and we can kind of take, collapse them, discover them through these different biclusters, these subsequently discovered biclusters, and collapse them. I don't think you'll, get something as simple as what I showed there, but that's more of a visualization. Thinking about making it more real.
Yeah. I second question. I work on co-expression networks, so I appreciate you mentioning that. And I'm wondering if
you can teach me a lot.
You have a lot of DNA sequence data, and I was wondering if it's much harder to get the expression data as well to look at for especially phenotypes.
Okay. Yeah, that's a good point. For the butterflies, I would say yes. I would say that will be more difficult. If we want to keep them alive, which typically we kind of do. You can, we can take legs and wing clips and they'll be okay. That might be a little more tricky, but is worth doing. I mean, certainly, and if individuals are, deceased and we can catch them right away, that might be really valuable to do. That's actually an excellent point. Okay, next question, yeah.
I wanted to ask you about the style or way of reproducing. So to get numbers, the family bringing a family is very efficient.
Yeah.
To get continued genetic flow, bringing one individual per family might be the way to go. So I wondered if you guys are recommending a change there, or if there's something else.
Yeah, so they try to, and that's another thing that, I mean, they had shifted to the model of the way they were doing translocations some way midway through. And to be honest, I would have to remember because I swapped them around. What was the change? So I should say the dispersive sex, the way that these scrub jays kind of operate is they have these family groups, and the males will eventually, a male who was a helper will butt off his own territory. And the dispersive sex is females. And so, yeah, possibly translocating a female in would emulate a more natural kind of, that sort of dispersal. I think the idea has been that survival rates, if they do, they're just much more able to survive if you move the families in. So that's kind of the rationale there. And I've had I think it's interesting, I've had a lot of interesting ideas about what you could do. I like presenting this because I like the ideas of how to pull this off, and maybe avoid this sort of situation. One idea that I really liked was, if it was possible actually like cross-foster or foster eggs, introduce eggs. And the scrub jay folks really don't know if they'll reject. But that could be a cool way to incorporate a lineage into an established family where maybe they would have higher survival. But the scrub jays are pretty savvy, so I don't know if you could pull that off. But it's something, consider. All this is that require a lot of work and it's challenging. But yeah, at this point, I think there's been in favor of moving family groups just based on their odds that they'll survive, and they want. Given that they're taking birds from these kind of threatened populations, they're kind of hoping, they don't want it to be for naught.
Yeah, that was super fun. Really enjoyed all of that. I was curious looking at the reproductive skew for scrub jays. Has there been enough generations, if you have enough power to try to see which bits of T171 are being transmitted? Is it is it a sweep? Is this like a good thing for population, or is it conditioned on the pedigree, the amount of genome of that individual representing contemporary individuals? Is it unusual, or the distribution is unusual?
Yeah, understanding. That distribute. I mean, that's a great advantage of having the genomic data, right? That would be the big advantage there. And the only reason why I didn't jump in right away is because of the small time, the small number of generations. But that's not to say that it's not worth doing, and is on the to-do list is look at that. But so I don't know. I imagine the I kind of suspect it won't mean a lot. Understanding that it'll be hard to get any takeaways because of the short numbers of recombinations that we could get over this amount of time. But yeah, is the thing that would be interesting to monitor. Certainly, continuing to look at through these temporal snapshots, definitely.
The natural history of the jay. What makes this individual that is overrepresented now in the population that makes it more successful? Do you think it's something
specific about their behavior or size or whatever it is, or is something more random?
Yeah. About the particular individual, I don't know. And that's what we would sort of like to see. That's why we're digging into all the demographic data, seeing if there's really something that aligns with these highly successful Jays in there. That's also on the to-do list. So I don't know, and I don't think, to my knowledge, people understand really what is inherently making any particular individual particularly successful. But we have the data, and now we can look at that. And again, because we have the genetic data, that's important because again, we could compare that Texaco bird that looked like a high contributor and maybe get the wrong impression. But now that we know is actually leaving a genetic footprint, now we should be able to reliably do that from the demographic data. That confounding effect.
Quick follow up to that. How confident are you that 71 was actually that individual?
Yeah.
Because I noticed the other two green ones were introduced very early.
Yeah. The other two individuals that were highly he was introduced with the family group, and there was those other individuals that we don't really see a lot of their ancestry in that population. So again, I can't be certain that because we don't have every translocated bird sequence. We have 30 of the 51. Above the ones that we have, just the way that skew parameter is estimated, it's like that whichever one has, if they have to be more related than any of the other ones, so they are the most kind of likely direct ancestor. But that's not to say, I think there was maybe, I'd have to look if there was more birds from site 1 that could have actually been, a sibling of that bird or something like that. So I wouldn't say 100% confident, but yeah.
I was just wondering, do you know if this type of skew, the genomic contribution of the population from specific individuals, is this something that you see often in nature, like how the population perform? What do we know about the baseline sort of genetic or demography of healthy populations, and is this informing your work on this
Yeah, I think one of the things that is leading to the high skew here is this, of course, it's this demographic the population's small, and so there's a chance for just a small number of individuals to fuel, when the population is very small, I think the chances become higher for, very small individuals to really fuel this expansion. That's not to say, that there hasn't been plenty of natural founder events that, have resulted from the same thing. And if you look at those populations now, what they look like, if, the ones that did have that happen, we don't detect because they all went extinct, or if they're there and, that is the picture. But I haven't really done this analysis in many other populations, and I don't know myself what that looks like naturally.
Yeah.
I'd be open to learn.
Yeah, I was wondering if the social structure has something to do with that as well, because you mentioned the wolf example.
So that's what I mean, and that's a good point. That kind of brings me back. To the fact that, like I said, I think, just given how the dominant personality of these birds, their territoriality, like I was saying, I think it kind of could be the null model of animals with this kind of life history, with this kind of behavior. And it just really brings me back to think that the reason that was a valid evolutionary strategy is just because you had historically more gene flow, which now just doesn't work in a very fragmented world.
Well, it's already our bedtime, I guess. So join me in thanking Tyler for an awesome talk. And if you want to hang around for the reception,